Good morning and good afternoon, everyone. This is Alexis Myers from the IQ Consortium Secretariat. I would like to thank you for joining this morning's IQ webinar on QSP as an integral component of clinical development, case studies presented by the IQ CPLG QSP Working Group. This webinar will be recorded and will be made available via the IQ Consortium website following today's session. Before we begin the webinar, I would like to take care of a few housekeeping matters with you. Firstly, the webinar presentation and subsequent question and answer session will conclude no later than 12 noon Eastern Time. Secondly, you will see a panel on the right-hand side of your screen. Clicking the arrow shown here in the green box will allow you to collapse or expand this panel. Next, VoIP participants using your computer speakers can avoid audio feedback by using a headset and keeping wireless devices away from their computers. For those dialed in by telephone, please enter your audio pin shown on the screen in the audio pane of your GoToWebinar panel. And finally, a Q&A session will take place at the conclusion of the presentations today. Questions may be entered in the box highlighted in red here during and after the presentation. And I would also just like to briefly provide you with our customary antitrust admonition that this webinar is subject to antitrust guidelines adopted by the IQ Consortium Board of Directors. These guidelines require us to stay away from discussion of any commercially sensitive topics such as pricing or market shares. And part of my role on the teleconference today is to make sure that these guidelines are adhered to. So with that, I'm going to hand things over to the moderator of today's webinar, CJ Musanti.
Thank you, Alexis. Hello, everyone, and thank you for joining today. My name is CJ Musanti, and I'm a research fellow and head of systems modeling in Pfizer's Internal Medicine Research Unit. And I will be the moderator for today's session. The Quantitative Systems Pharmacology, or QSP Working Group of the IQ Consortium Clinical Pharmacology Leadership Group, so that's the CPLG, consists of QSP modeling engineers, PK/PD scientists, clinical pharmacologists, pharmacometricians, biologists, and clinicians. And this was a vast group of researchers that was chosen to represent the various functions that contribute to successful QSP modeling efforts. In addition, 11 IQ Consortium member companies are represented, plus 2 QSP modeling CROs. For today's session, we'll have 5 speakers, each in a different geographic location and also with a different job function. Each speaker brings a unique perspective to the discussion, so we do ask that you please have patience with us as we transition between slides and speakers. So with that, it's my distinct pleasure to introduce our first speaker, Dr. Pete Zondergraaf, who will provide an introduction to the topic. Pete?
Thank you, CJ. Welcome, everyone. Can I have my first slide, please? In the last 2 decades, there has been a steep growth in the application of modeling and simulation across the pharmaceutical R&D spectrum, which now ranges from modeling of large biological pathways to guide the identification of new drug targets to the prediction of cost effectiveness using pharmacoeconomical models. In drug development, modeling and simulation has become an integral and expected part of internal and regulatory decision making, as illustrated by regulatory and industry best practice guidelines. For example, the European Federation of Pharmaceutical Industries and FPA recently published a good practice document which covered all aspects of planning, conduct, documentation of quantitative approaches across R&D, which they labeled Model-Based Drug Discovery and Development, or MID3. In this white paper, the value of MID3 and impact is evidenced by some 100 case studies. For example, Pfizer reported that model-based drug development approaches enabled a cost saving in the clinical trial budget of approximately 100 million and resulted in an increase in late-stage clinical trial success, whereas Merck reported cost savings of $500 million through MID3 impact. Next slide, please. But however, despite this clear impact and the return on investment of MID3 across industry, there remains a significant opportunity for a broader and deeper impact. In particular, the attrition in late-stage clinical development remains high. Lack of efficacy or differentiation is by far the main reason for failure in late-stage drug development. For example, in a recent analysis, Harrison estimated that lack of efficacy was the reason for 48% of failures in phase 2 and 55% in phase 3. Also, the FDA recently published some 22 case studies where positive clinical trial results in phase 2 did not translate to phase 3. Next slide, please. One of the fundamental origins of such failure is a lack of quantitative understanding of the complexity of disease biology in diverse patient populations, which makes the accurate prediction of the impact of perturbing such complex systems with a drug very difficult, in particular with quantitative approaches that are largely empirical. Therefore, some 10 years ago, quantitative scientists from industry, academia, and regulatory agencies started to focus on bringing together the disciplines of pharmacometrics and systems biology, which until then had operated more or less independently. This resulted in the publication of this white paper, sponsored by the National Institute of General Medical Sciences in 2011, which is widely seen as the milestone defining the birth of the discipline of Quantitative Systems Pharmacology, or QSP, as applied to drug discovery and development. In the white paper, QSP is defined as an approach to translational medicine that combines computational and experimental methods to elucidate, validate, and apply new pharmacological concepts to the development and use of small molecules and biological drugs. Next slide, please. The schematics in this slide are from 2 recent papers, illustrating that QSP can be regarded as an approach to integrate features of the various modeling approaches, spanning the spectrum between bottom-up systems biology and top-down pharmacometrics, as Rick Burtz will further explain in the second part of the introduction to the webinar.
Thanks, Pete. Hi, I'm Rick Burtz, and I'm the head of clinical pharmacology and pharmacometrics at Bristol Myers Squibb. My group also includes a QSP group. I'm going to talk about the role of QSP in clinical development, really looking at in a little bit more detail at QSP versus pharmacometrics or PK/PD. This slide here shows a schematic of really the evolution of the systems pharmacology from PK/PD or pharmacometrics analysis. If you look at really how you might consider the two, QSP really could be considered an extension or evolution of pharmacometrics. As noted in the schematic, if you look at the right, you'll see that PK/PD uses the majority of cases, in the majority of cases, model plasma concentrations or population PK derived from a drug dose, and then that data is modeled using generally a statistical approach to explore relationships of exposure or disease severity, safety or efficacy parameters. Additionally, covariates can be added, such as disease, severity, patient subtypes, demographics, and concurrent meds. They can be incorporated into the model as well. In contrast, QSP actually is more mechanistic in nature, and if you look at the the schematic here over on the left, that would be expanding efficacy into a QSP model, which links biology, physiology, and pharmacology to endpoints of interest using a systems approach. Really, if you look at the figure on the left, what that represents is just an example, and it's really a systems model exploring the immune system, where specific effects of a drug on T cells, and thus a clinical endpoint such as tumor shrinkage, can be explored. Just want to reinforce that both modeling approaches are complementary to each other. Next slide. This triangle is another way of representing what is called here integrative drug disease modeling. And if you look at parts of the triangle, each represents a portion of what would be done or what could be done in modeling. Really, the top portion represents the comparative clinical trial data, actually, where efficacy points are compared across compounds. And the other middle part is the PK/PD modeling or pharmacometric modeling, which really is represented here as a top-down approach. One generally needs clinical trial data in order to perform the modeling and simulate outcomes. At the very top, as I mentioned, was the comparative clinical trial data. A quantitative systems pharmacology model, which is represented at the bottom of the triangle, can be used to simulate outcomes without clinical data from a specific drug, building the model with primarily extensive available literature data on the biological system and in vitro experimental results. This would be called more of a bottom-up approach, if you will. QSP models can predict the outcome from administration of a particular drug with a known mechanism, compare drugs with a similar mechanism, and drug combinations for the same indication with differing mechanisms. All the information that's obtained from PK/PD and other human trials, all the way up the triangle, can be incorporated to strengthen the predictive accuracy of the QSP model. Next slide. This slide really refers to the role of regulatory science and decision-making for QSP. And this is really, I think, in its infancy, but we are seeing some examples of interest in using mechanistic models for evaluating potentially packages, I'm sure that we will see QSP models submitted as part of registrational packages. As noted previously, I mean, most pharmaceutical companies, they've already incorporated QSP into their drug discovery and development, as evidenced by the number of companies we have in our working group. An article that was published this year, earlier this year by Abernathy, illustrates where QSP may fit, and that's over on the left-hand side, as a tool in the drug development continuum from the regulatory point of view. It's viewed that the use of mechanistic models may significantly enhance model-informed drug development, and there is even a discussion of the value of building pre-competitive QSP models for varying diseases to help with development of drugs and analysis of drugs and regulatory packages. The paper on the right-hand side by Mark Peterson, who we happen to have as part of our panel, describes for the first time a QSP model that was used by the FDA to inform an NDA review, and Mark will elaborate further during his case presentation. Next slide. There will be 3 case examples today, and these are really, again, looking at QSP as an integral component of drug development. Each of the cases are quite different. They each illustrate QSP and what role they played in drug development. But the cases cover different stages of drug development. It could be anywhere from preclinical to phase 4, and in different therapy areas, cardiovascular, endometriosis, oncology, and from 3 different companies, AstraZeneca, Pfizer, and Genentech. I'd now like to turn it over to Gabriel Helminger, who is going to present the first case, a cardiorenal systems model in support of CV and CKD drug development. Gabriel?
Yes, thank you, Rick, and welcome everyone. My name is Gabriel Helminger. I'm responsible for translational modeling sciences within Quantitative Clin Pharm at AstraZeneca. It's a pleasure to present today a systems model of cardiorenal physiology and pharmacology. This model has a long history of development and applications. And I'd like to acknowledge Melissa Hallow, today at University of Georgia, Sergei Ermakov, today at Amgen, and also Anna Kondic, today at Merck, who were all part of the original model development team. On the next slide, the case study deals with inhibitors of sodium glucose transporters and a model-based analysis of the potential benefits towards renal and cardiovascular protection, which this class of compounds may provide. But first, I'd like to mention previous applications of this model. It was initially developed for targeted questions around hypertension and kidney disease programs in phase 2, phase 3. And that's the first point I'd like to emphasize about the systems models. Whatever they represent around the biology, physiology, and pharmacology, they are initially developed for specific questions which have been scoped within project teams, so-called fit-for-purpose modeling, as Rick has just mentioned. This model was initially developed for targets around the renin-angiotensin-aldosterone pathway and effects of its modulation towards hypertension and kidney disease treatments. In the end, we supported as many as 5 targets, 8 drug treatment modalities, including combinations in various patient populations. And a second point I'd like to make about these systems models is while you initiate their development based on drug development questions, at the same time, you may want to have a sense of whether they may be reused pending further adjustments for related mechanisms, drug targets, questions on dosing, drug combinations, or patient selection. And this is exactly what happened with this hypertension and kidney disease program I'm referring to. We initially did not face that many targets or compounds to address. We were focused on a phase 3 program initially. But we knew we had a rich pipeline of compounds and potential combinations coming up to be addressed with a companion systems model of this type. So more recently, we added the mechanisms of SGLT2 inhibition into the existing cardiorenal modeling framework, and I will describe now how we developed and used the model for that purpose. So on the next slide, the questions for this case study focused around the emergence of data on empagliflozin, an SGLT2 inhibitor, in the EMPA-REG cardiovascular outcome trial, where the drug showed indeed benefits towards cardiovascular outcome. And that included an impressive 30 to 40% reduction in heart failure-related hospitalization, which you see on the graph on the left-hand side. The drug also provided renal protection, as you see on the right-hand side, with a slowdown in the long-term decline of the glomerular filtration rate, despite an initial drop in that rate. One feature to notice up front on this graph is the very fast separation of the curves versus placebo, literally within first week of treatment, and we'll get back to this later. So looking at this finding, one may wonder, are these findings relevant to SGLT2 inhibition? Meaning, is it a class effect? And if so, what would be the mechanism driving these outcomes observations? And if it's a class effect, how would one go around designing outcomes trials? What kind of patients should we enroll? Diabetics, non-diabetics, what level of renal impairment for these patients, which dose to use, and so forth. So on the next slide, indeed, this slide shows the different compounds within the cardiorenal model. And to be clear, we did not develop this model from scratch. We actually borrowed from existing physiology and models by Arthur Guyton in the 60s and 70s, and others as well, to which we added extensions of target biology and pathophysiology. At this stage, in addition to the in vitro and in vivo data we also used clinical data from about 31 trials representing around 21,000 patients to test and calibrate the model. In terms of a quick guide to the model, we may start at the top left, which describes the renal vasculatures, where you have inlet and outlet pressures. From there, you can back calculate pressures along the renal vasculature. This then becomes an important step to determine glomerulus pressure, oncotic pressure, Bowman space pressure, all represented in the nephron, which you have on the lower left graph. And from there, you continue calculating glomerular filtration rate, which feeds into the tubule and allows fluid to move along the tubule, carrying water and sodium ions, for example. A small amount of that filtrate is then reabsorbed as water and sodium, which feeds into the cardiovascular module on the top right. That module, featuring cardiac output, blood pressure, then closes the loop back to the kidney. Now, at steady state, you can see how this would represent a straightforward plumbing system. However, the entire system is actually highly regulated with multiple regulatory mechanisms, ensuring the system can always go back to homeostasis. So in the lower right corner, you see these regulatory mechanisms, which we represented in the model. It includes features such as tubuloglomerular feedback, renal interstitial hydrostatic pressure, vasopressin, and other mechanisms. For example, if you look specifically at the afferent resistance block, you see how the model captures the regulation of that block by tubuloglomerular feedback, myogenic autoregulation, and the RAS signaling system itself, which further modulates other physiological effectors on that cartoon. SGLT2 as a target, and whereby the PK or the dose comes in as well, is not shown on these diagrams explicitly, but it is explicitly expressed, for example, in the effector function eta-PT on the lower left cartoon, which describes sodium reabsorption. So moving on to the next slide, equipped with the model, we wanted to explore different mechanisms which would lead to the benefits observed in EMPA-REG. This slide summarizes plausible mechanisms leading to such benefits. Now remember, in the EMPA-REG study, my previous point, you see very fast separation between treatment and placebo groups, literally in a matter of days. And some of the proposed mechanisms in this diagram, for example, the gray shaded area on the left-hand side, where you see decreased body mass, inflammation, fibrosis, increased cardiac contractility, all these mechanisms will simply not work on such a short time scale. For fast separation in the outcome study, to achieve that, we started to think about a hemodynamic effect, which could set in more rapidly upon SGLT2 inhibition. So using the model, we actually investigated the hemodynamic mechanism you see on the right-hand side in that other gray shaded area through the model, and we showed that this mechanism, in fact, while reducing indeed intraglomerular hypertension and possibly decreasing renal events in the long term, in the model we showed that this mechanism would result in more rather than fewer renal events in the short term, which is not what you see clinically. So moving on to the next slide, we pursued other hemodynamic hypotheses using data from a recent clinical pharmacology study shown on the lower panels. We first hypothesized that the SGLT2 inhibition would have only direct effect, meaning reduced sodium reabsorption only in the proximal tubule, as shown on that upper left cartoon. This would be similar to a classic diuretic effect. We first set the magnitude of SGLT2 inhibition effect to correctly reproduce the urinary glucose excretion data that you see on the first of the 6 mini-graphs. Then we ran the model forward. Under such an assumption, however, you cannot reproduce the data shown on the other 5 mini-graphs on that slide. So what we did next, we assumed SGLT2 inhibition to have both direct and indirect effects, meaning decreased sodium reabsorption, but also decreased glucose reabsorption in the proximal tubule. So in the model, this then resulted in an increase in osmolality, which then drove the osmotic diuresis effect, whereby you effectively inhibit water reabsorption. That mechanism now is very distinct from a classic diuretic and is actually necessary to reproduce all the data you see on the 5 mini-graphs on the lower part of the graph. So next slide. With this plausible mechanism of direct plus indirect effects of SGLT2 inhibition, we explored what happens at higher physiological effector levels. And the model here shows that the induced osmotic diuresis, uncovered on the previous slide, that further results in fast and sustained reductions in extracellular fluid, or ECF volume. That's a physiological feature we can simulate within the cardiovascular function of the model. This sustained reduction in ECF volume actually occurs with minimal reduction in blood volume, which is a desirable feature in heart failure patients. And the 4 mini-graphs on this slide illustrate this with a direct inhibitory effect on sodium reabsorption only. You actually see a transient reduction in blood volume and not much of an effect at all on ECF volume. But now if you add the indirect effect, you get this fast and sustained reduction in ECF volume, while blood volume again goes back to nearly to baseline. And once you add, the indirect effect of glucose reabsorption inhibition, you get this fast and sustained reduction in ECF volume. So that's a very important feature here. We actually performed this model-based analysis for both a classic diuretic, such as bumetanide, versus an SGLT2 inhibitor, and that's shown on the picture insert on the top right. And we found that the ratio of changes in ECF versus blood volumes, that ratio is more than double for SGLT2 inhibitors versus bumetanide. So this fast and sustained reduction in ECF volume appeared to be a mechanism also supported indirectly by rapid weight loss data in healthy subjects. So of all the hypotheses we explored, and with the clinical data at hand, ECF volume reduction was the most sensible one to explain this fast-setting renal and cardiac protection benefits. So on the final slide, here you see we now added our proposed mechanism on the left-hand side to the literature diagram shown earlier on. And to conclude on this first case study, we use a model of cardiorenal system physiology and pharmacology to support the generation of a sensible hemodynamic-driven mechanism whereby SGLT2 inhibitors may provide renal and cardiac protection through a fast, sustained reduction in ECF volume. Next, having calibrated the magnitude and the kinetics of this ECF volume reduction, and using recent clinical pharmacology data, we can now use the model to predict the effect for specific patient populations whose characteristics can actually be set in the model, and of course for different dosing regimens of specific SGLT2 inhibitors. So using a systems model and together with other pharmacometrics and also model-based meta-analysis we performed, we gained much more confidence in the design of future outcome studies. So from here, I'd like to hand over the baton to Mark Peterson, who will next present a systems model of bone to address a specific problem on safety versus efficacy. Thank you.
Thanks, Gabriel. Hi, I'm Mark Peterson, Director of Global Pharmacometrics at Pfizer, and I've been involved with QSP modeling for a number of years at various places. The case I'm going to highlight is actually two pieces. The first looks at the combination of a QSP model with a model-based meta-analysis to evaluate a drug's therapeutic index and lead to a program decision. The second looks at the same QSP model used in a regulatory setting by the FDA to assess a new drug application in the absence of dose-ranging information. But before I continue, I have to emphasize that the work I'm showing here are extensions by others of a model that I initiated with Matt Riggs and we first published in 2010. So I'd like to be sure and thank the numerous individuals who contributed both directly and indirectly to the materials in this talk, and all those who have added, published, and presented extensions of the bone and calcium model over the years, especially Matt Riggs. One related takeaway that you heard from Gabriel is that repurposing an extension of QSP is a key value feature of these models, along with their ability to inform drug development decisions. So for the first part, let's look at how a QSP model was combined with an MBMA, or model-based meta-analysis, derived from published response data to gain insights into efficacy and adverse event potential for a compound. The disease in this case was endometriosis, and the drug mechanism was GnRH modulation aimed at reducing the endometrial symptom severity score, or ESSS. And the goal of the work was to inform the design of a trial so that it could read out quickly and provide insights into both efficacy and adverse event potential. So since the key to affecting endometriosis is modulation of estrogen, and estrogen is implicated in bone homeostasis, the research challenge was to identify a GnRH modulator dose that would give the desired improvement in ESSS and an acceptable level of impact on bone mineral density, or BMD. But since BMD leads to a required longer length and size of trial due to its variability, the trial design was a concern. Because of the variability and the longer trial, the idea was perhaps there was a way to use QSP and MBMA to look at various biomarkers that might change rapidly and provide known information about the expected outcomes, but with a shorter trial. If a quickly responding biomarker could be identified that was known to be related to the physiology and linked to BMD, that would allow time to be saved and money to be saved and to get quantitative readouts that were important to making the decision to move forward with subsequent trials. So the approach was to use the known relationship between GnRH modulation, resulting estrogen levels, and ESSS impact from the literature to understand, based on some early PK/PD for the new compound, what the expected joint outcome of BMD and ESS would be predicted to look like, and what biomarkers, more importantly, would be best as earlier indicators of that outcome. So let's see what this looks like in terms of the models. On the left is the QSP model of bone and calcium homeostasis, which provided the core platform. It included the major organ systems implicated in bone and calcium homeostasis: kidney, parathyroid gland, gut, bone, and vasculature, as well as estrogen and its effects on BMD. So therefore, it was sufficient to address expected effects on BMD when estrogen was affected through the GnRH modulation. There were a number of different GnRH modulators with published data, those are shown in the top right, that provided input on the degree of estrogen modulation they produced and the impact that would be expected on lumbar spine BMD. And that's shown here as a couple landmarks with the 0% BMD change shown as the dashed line and a reference of a 2.2% loss or change in BMD indicated by the dotted line. The solid line is the QSP predicted BMD seen with various reductions in estrogen, which is on the x-axis, produced by GnRH modulation. And as you can see, this indicates that the QSP model produced a solid prediction of expected BMD changes with differing degrees of estrogen modulation. The papers also provided predictions of endometrial ESSS that allows the changes in estrogen with a new compound to be tied back to the predictions of ESSS. Having these elements, it is then possible to predict probability of taking a patient from the most severe ESSS status 4 and reducing their pain status, as shown in the lower right panel with the green circles, as well as the probability of a patient becoming pain-free with decreases in estrogen, as shown with the red line and squares, while simultaneously evaluating the anticipated percent change in BMD, shown by the blue line with triangles. Also, the QSP model, because it generates the biomarkers related to physiology simultaneously, it was possible to determine that estrogen, not the bone resorption or formation markers, as might have been thought, changed rapidly enough with treatment to make it a reasonable early indicator of a long-term outcome. And what can be seen is that with estrogen levels between about 20 to 40 picogram per mil, one can achieve an increased response without dramatic changes in BMD. However, there still needs to be a decision as to whether the therapeutic index was adequate to move forward. And that was the next step once the modeling was complete. The conclusions from this work were that estrogen would be the most useful early indicator of response. However, it was a feeling that the variability in BMD might lead to some individuals having too great a bone mineral density loss over time. Therefore, the inability to separate endometriosis efficacy from the associated adverse event of bone mineral density loss due to the variability in BMD led to a decision not to continue to invest in GnRH modulation. The impact, as you can imagine, was all future investments in GnRH compounds for endometriosis was halted, and the resources were able to be redeployed. The second part of this case is the reuse of the bone and calcium homeostasis by the FDA to assess the NDA of Natpara, a PTH analog for use in control of hypocalcemia in patients with hypoparathyroidism. To the best of our knowledge, this is the first time that the FDA has used the QSP model to assess a new drug application. In 2014, in an advisory board meeting, they presented their use of the model. The reason for using the QSP model is described well within the advisory board materials and hinges on the need to simultaneously model and understand multiple endpoints that are physiologically linked to the therapeutic in question. In this case, PTH, vitamin D, and calcium in both serum and urine. The nice part about having the bone and calcium homeostasis model available was that there were minimal changes needed to the model to get at these questions and the answers that they wanted, in the form of the addition of the PK model components for Natpara, vitamin D, and a urinary excretion compartment to estimate that value. Specifically, the agency wanted to use the bone model to address whether there were alternate dose scenarios and release profiles that would result in drug levels closer to physiologic and therefore better control of hypercalciuria than what was observed for patients dosed with the proposed 100 microgram QD regimen. The overall approach to applying the QSP model to this work was simple in concept. Nab-paro met its primary endpoint in the studies, so that really wasn't a concern. But there had been no apparent control of hypercalciuria demonstrated. And since the PK of nab-paro didn't mimic the physiologic profile of PTH, and pharmacodynamic effect on reducing calcium excretion was short-lived, with serum calcium levels dropping below normal and urine levels remaining elevated within a 24-hour period. So this was a bit of a problem. Also, since there was no dose-finding study in the dossier to indicate whether other regimens might lead to better control of hypercalciuria, there was no way to compare alternate doses or regimens to assess the findings. So the plan was to use the QSP model to fill that void and evaluate alternate dose regimens and the impacts on serum and urinary calcium simultaneously. Quick note on validation, as this is often a question that comes up around QSP models due to their complexity. As can be imagined, for use at this level, one would want to understand how robust the model is. To address this, the FDA evaluated this via a fit-for-purpose validation scheme. This is fairly simple in concept and provided a level of confidence that added to the published uses of the model. Matt Riggs and I were also delighted to see the validation, as it was done completely independently from data that we had used to develop the model. And so the schema is shown here, and simply put, use data from the filing to confirm that the QSP model, without structural or system model parameter modifications, except those needed, as we discussed a moment ago, was able to describe the observed data adequately and within acceptable limits to give confidence that the model could be used for the problem at hand. And if you were to go online and pull down this data, you'll see their validation, how they went about validating this and some of the graphical outcomes. Having demonstrated the suitability in this way, the FDA moved on to assess alternate regimens and formulations. There are a number of regimens and doses that were explored by the FDA and included in the appendices online with the QSP model and those. I guess I should stress that this is a valuable feature that one can in a very short amount of time, look at a lot of scenarios and see how the physiology responds comparing multiple outputs simultaneously. In the interest of time, we'll just look at this one. On the top figure is shown the PK of the 100 microgram QD dose in red, giving largest peak, a 50 microgram BID dose in green, giving 2 peaks, and a 50 microgram QD slow release formulation in brown, with the gray area indicating the normal physiologic range for PTH between 10 and 65 picogram per mil. On the left lower panel is the predicted calcium levels achieved for these 2 alternate regimen formulations, as well as the original for the 100 microgram on the bottom. And on the right is the predicted urinary calcium for each of these. As you can see, the QSP model predicted that these 2 alternate regimens would provide suitably improved serum calcium control with respect to normal clinical ranges and improved 24-hour urinary excretion level of calcium, lowering the possibility of generating kidney stones and manifesting adverse events. So what was the outcome of this exercise? Well, the FDA didn't change the sponsor sought approval. The QD dose regimen was approved, but the titration of doses was indicated in the label. At the same time, in the absence of the dose-ranging information, the QSP model analysis supported future investigations of nab-paro regimens in a post-marketing setting, and the sponsor committed to 2 trials being conducted evaluating the effects of dose and regimen on hypercalciuria that should read out in the near future. With that, I would like to thank all of you and hand off to the next speaker for case study 3.
Okay, thanks, Mark. My name is Wayne Chu. I'm a Senior Medical Director at Genentech. And for this case study, I'm going to talk about how QSP modeling has been very important in informing the activities of an ongoing phase 1 study, specifically the phase 1 study of the anti-CD20/CD3 bispecific antibody that's currently in development for the treatment of non-Hodgkin's lymphoma. Before I go on, I do want to first and foremost acknowledge the collaborators who worked to develop and implement this model and has really done a fantastic job working with the team in order to help maximize the potential of QSP modeling to make important clinical decisions. And I do want to point out Iraj Hasani, Chichong Lee, and Saroja Ramanujan as being the principal architects of the QSP modeling effort. Next slide, please. So just a little bit of background on the molecule. As everyone's aware, cancer immunotherapy is becoming an area of extreme interest in drug development, utilizing a patient's immune system for the treatment of cancers. Among these different modalities, the use of bispecific antibodies, whereby an antibody is adapted to recognize simultaneously a tumor target and an immune cell, this particular therapeutic platform is of significant interest. For the CD20, CD3 bispecific, this is a fully humanized immunoglobulin monoclonal antibody where it's composed of two half antibodies. One half antibody binding a tumor target, in this case CD20 on the B cell, and on the other side, targeting CD3, which is a component of the T cell receptor on T cells. And so this is a molecule that was produced by a knobs and hole technology. And as you can see by the diagram in the bottom, the mechanism of action of these class of molecules is fairly straightforward in that it recruits T cells to be in close proximity with a target tumor cell. And that close approximation results in the formation of what's called an immunologic synapse, which leads to the activation of T cells, the release of cytotoxic granules that ultimately result in killing the tumor cell. Next slide. So there have been a number of these types of molecules in development, and this includes not only the bispecific molecules, but also the development of other T cell-based therapies such as the CAR T. And what we know from these molecules is that they're not only highly effective in terms of being able to kill tumor cells, but they also carry certain safety liabilities that are a result of immune activation. And it's really these safety liabilities that are important determinants in identifying the optimal dose and schedule for these molecules. So in the case of T-cell recruiting molecules for the purposes of treating B-cell malignancies, such as B-cell lymphomas and leukemias, the principal toxicities that have been described are cytokine release syndrome and neurotoxicity. So with respect to cytokine release syndrome, this usually manifests as flu-like symptoms such as fevers and chills, but when you have an uncontrolled cytokine release, that can lead to very severe clinical effects, including hypotension, respiratory failure, and end organ failure. And in rare cases, this has led to patients dying of this immune-mediated complication. The other toxicity associated with these class of agents is neurotoxicity. And this clinically manifests as confusion, aphasia, which is the inability to express or understand language, tremors that can range from focal tremors to frank seizures, and encephalopathy, which also has resulted in a number of deaths with this class of molecules. And as I mentioned, while the mechanism of both is not fully defined, they are both believed to be associated with immune activation following administration of the agent, be it bispecific or a CAR T. So the fundamental question and an open question with this class of molecules is, can the bispecific be administered in a way that reduces the risk or ameliorates treatment-emergent toxicities? Because you can imagine that, especially as you move these molecules into patients with newly diagnosed disease, there's more of a premium on attaining, efficacy, but while maintaining a very safe profile for the agent. Next slide. So when we developed the bispecific and was trying to develop ways to dose the molecule in patients, we did have some clinical precedents in the form of blinatumomab. So blinatumomab is a bispecific with a different structure. It targets a different antigen on B cells, but the mechanism of action, remains the same. And blinatumomab has been in development for quite some time for the treatment of acute lymphoblastic leukemia and non-Hodgkin's lymphoma. And we've had the fortune of being able to leverage some of that clinical experience with respect to making our own dosing decisions. But as you can see from the blinatumomab experience, what ultimately resulted in the ability to dose to a higher dose level was the implementation of step dosing. Step dosing in this case just means that you start off with a low dose and you gradually increase the dose over a period of time to your target dose. And as you can see by the table in the upper right-hand corner, this was a dosing schedule that was implemented for blinatumomab based on their ability to mitigate against neurotoxicity. And as you can see in the figure in the bottom, that by implementation of the double-step fractionation, that mitigates the magnitude of the cytokine release. If you look at the figure on the left, where the arrow is, you can see that the step fractionation, while it did result in elevations in cytokine levels, that level of cytokine increase was substantially less than the cytokine level that was achieved when you did not give the drug as a step fractionation, but you just gave it as a target dose, as illustrated on the right. And so clinical examples such as these were important clues for us on the CD20, CD3 bispecific as a way of trying to dose this molecule. However, one couldn't just simply extrapolate the clinical experience of blinatumomab for our molecule, primarily because of the very distinct PK characteristics, the difference in target, and the fact that because of the PK, blinatumomab is given as a continuous infusion, whereas for the CD20, CD3, because it's a full-length bispecific antibody, the intention was to dose intermittently. Next slide. So as a consequence of this, this is when the decision was made to implement QSP modeling as a way to model the B and T cell dynamics in the presence of a bispecific. And so this involves a lot of different pieces, including the fundamental knowledge of or a synthesis of what we know about the immune system, and not only how it works, but what is the trafficking and dynamics with respect to the various immune populations that are important for this class of molecules to work. B, preclinical data obtained from our own experiments. And C, clinical data obtained from other molecules such as blinatumomab. And what you can see is that all of these aspects feed into the QSP model, which is illustrated in the middle of that figure. And it takes into account what we know about what happens to T cells when they're activated, what happens to target cells when they're destroyed, and so on and so forth. And so the mechanistic model is really based on what's the known mechanism of action, as summarized in those bullet points below, where T cells are activated in the presence of local target B cells, and they subsequently produce cytokines. We use IL-6 as a surrogate of cytokine release. This has been demonstrated not only biologically, but also clinically. And then thirdly, multiple relevant compartments. So we know that the immune system has different dynamics with respect to whether or not they're in peripheral blood, spleen, lymph nodes, bone marrow, and tumor. And so we integrated all of these different aspects to generate the QSP model, and then validated and predicted using a variety of different data sets. So for the preclinical development validation, this was based on preclinical data of the bispecific obtained in cynomolgus monkeys. The clinical validation was based on the prediction of published blinatumomab data in acute lymphoblastic leukemia, and then prospective prediction, which was implemented and leveraged using published blinatumomab data in non-Hodgkin's lymphoma. Next slide. So results of the QSP modeling, in essence, supported the use of double-step fractionated dosing for the anti-CD20, CD3 bispecific. And so you can see in the figure on the right an example of the types of results that were generated from the QSP modeling. And what you can see on the left are the various dosing schedules that were modeled, including a non-fractionated schedule, a cycle 1 flat fractionated schedule where you have equal doses of the bispecific given at those time intervals, and then thirdly, the step-up fractionation. On the right, the time course of cytokine levels, IL-6 levels. These are modeled results over time as a function of dose. And what you can see is that when you, depending on the type of schedule, the peak IL-6 level was quite different in that when the TDP was given on a non-fractionated schedule, you had the highest levels of IL-6, whereas when you move on to flat fractionated and then step-up fractionated, you see progressively lower peaks of IL-6, such that with the step-up fractionation, you see the lowest peak. And so it was for this, in part because of this reason, the decision was made to implement the step-up fractionation schedule in our phase 1 study. Importantly, one thing that we always concern ourselves with is whenever you try to blunt something like cytokine release, the other question is whether or not that has a deleterious impact on the anti-tumor activity of the bispecific. And so that was modeled as well. And the important result from the modeling was that no significant difference would be predicted in terms of anti-tumor activity. And so because of this, the modeling results, the phase 1 study, which initially started as a dose escalation with a non-fractionated schedule, has been amended to specifically assess the step fractionated schedule in patients receiving the CD20, CD3 bispecific. And while they're still early days with very few patients dosed, it does seem to reflect clinically with respect to the frequency and severity of cytokine release. And the hope is that with the step-up fractionation schedule, that we would not only be able to mitigate some of the treatment-emergent toxicities, but also try to achieve higher doses, which presumably would lead to greater anti-tumor efficacy. Finally, with respect to the QSP modeling, as far as this project is concerned, the model continues to evolve as we generate additional clinical data. And I think it's important to highlight the fact that the QSP modeling is not only important for the development of this particular bispecific, but you can imagine that the QSP model can also be used to support development of other molecules in its class, taking into account differences in the different mechanisms of action with respect to whether or not this is a solid tumor versus a liquid tumor, and then the different immune populations. And so, and finally, the model continues to be kind of updated to explore additional dosing strategies to mitigate toxicity while preserving anti-tumor activity. So with that, I'm going to move on and close out the presentation part of the session, just to highlight some major themes that I hope that you have heard through the presentation of these different case studies. I hope you will realize that quantitative systems pharmacology in drug development is a very iterative process, and it's limited and by the assumptions that go into the modeling. It's also limited by the amount of information that's fed into the model. But it's iterative in the sense that as you get more preclinical and clinical data, that model evolves and matures, and then in turn provides not only additional information that is important for decision making, but also provides additional information to generate additional hypotheses. And so the figure on the bottom is just to summarize that these informational inputs occur from both the preclinical as well as the clinical side. And they're all very interrelated, such that clinical information can inform preclinical decisions and vice versa. Next slide. And I think also what I hope is highlighted by these presentations is that the development and utilization of QSP modeling involves a lot of different people from many different disciplines. And this includes scientists, in discovery, all the way through clinical pharmacology scientists, and all the way through clinicians. And again, to emphasize that, QSP modeling is a rigorous, stepwise process. It's led by a team of scientists from very diverse disciplines. And the role of these individuals in totality help define the scope of the modeling effort, the model development itself, model calibration, qualification, and model application for hypothesis generation. Next slide. So just to recap, you've heard 3 different cases of where QSP has been shown to be an integral component of clinical development in all different stages of drug development, be it from late stage and post-marketing commitments all the way through ongoing phase 1 studies. And And again, I think you'll also see examples where QSP modeling can be applied across many different disciplines, many different disease areas. And as you can see, it's being implemented by a wide variety of groups and a wide variety of companies. So with that, I think the last slide is I do want to thank you for participating in the webinar. I hope it's been highly informative. And I just want to close out by acknowledging the folks that have enabled this webinar to take place. And this includes the IQ Clinical Pharmacology Leadership Group, Drinker, Biddle, and Reef LLP, and then finally the IQ Drug Metabolism Leadership Group. And so with that, I again thank you for your attention, and now I will turn it over for the Q&A session.
Thank you, Wayne. And so we are now going to open up the question and answer session. So I'll ask people, if you do have questions, to please complete them on the website as we have instructed earlier. But I think I'll start off now with a question to Rick. So through the case studies that we've shown today, we've seen the value of QSP to inform program decisions. But based on your experience at BMS, I'm wondering if you could elaborate on this and also comment on some of the challenges that you've come across in establishing a QSP group there.
Sure, thank you, CJ. Yeah, really in establishing a group, and our group at BMS is a little bit over 3 years old. I think that the key things that one needs to keep in mind, and challenges are, you need a good business case, and I think business case would obviously these 3 case examples are a good business case for why QSP is important, and also a strong advocate within the company, and certainly a leader you can identify to actually lead the group. I think building relationships with teams are important as you get started, and leveraging existing models. There's a lot of information out there, some CROs that you can use, and lastly, what's important to make the group sustainable is show results and impact, and do it with the so-called low-hanging fruit, or reduce models, or look at the questions that teams have that QSP really could answer. So that's, at least in my experience, I think what how to get a group started and how to avoid or how to really sustain it.
Okay, thanks very much, Rick. We do have another question on the line, and I think I'll direct this to Pete. Pete, do you think the QSP models developed for a disease area can be used to feed into the development of models for other disease areas? And what are the approaches that can be used for this repurposing?
Yes, thanks, CJ. So I think, yeah, the answer is yes. I think you've seen some very good case studies of models that have been used in specific indications, but actually, branching out into, slightly different indications. I certainly think with, for example, the current focus on immunology, that obviously, QSP models that contain elements of the immune systems, may be used in oncology, may be used in arthritis, maybe even Alzheimer's. So I think certainly there's scope for these models to be used in different indications and therapeutic areas. Yes.
Okay, thanks very much, Pete. There are also some specific questions for cases, and I'll present the first one to Gabriel. Gabriel, were the indirect effects of SGLT2 inhibition explicitly added as input to the model, or were they predicted by the model?
No, that was explicitly entered into the model. Yeah. So it's one of those knobs that you can actually modulate, and then you decide which knobs you want to act upon to see if it reproduces certain output data, which can occur at different physiological scales, cellular or more physiological. So yeah, that was explicitly represented and inputted into the model.
Thank you. I think it also shows a nice example of hypothesis generation of QSP models and how that can be used. We do have another question. This is for Wayne and Sureja, if you are also still on the line. In that example, how did the preclinical model translate the CD20 expression level? And I said, I assume that they are healthy monkeys. So
can you hear me?
Yes, you can.
Okay. Yeah, so what we did is we actually took into account, in translating from the cyno to the human, we took into account things like different cell numbers, different, if there was evidence of different potency of the molecule against cyno B cells versus human B cells, all of that was explicitly put into the model. And yes, it was a healthy cyno, and that was part of the reason for the validation against the of the translational approach with the blinatumomab ALL data, was to verify that our approaches to translating from a healthy cyno first to a healthy human, and then essentially virtually giving that healthy human a disease, in one case ALL, in another case NHL, would generate appropriate biological predictions. So that was verified that we could actually do that through the validation of the blinatumomab ALL predictions.
Yeah, and the only thing that I would add is that we do know that in terms of CD20 expression levels, they generally are fairly consistent across healthy B cells as well as malignant B cells.
Okay, thanks very much, Wayne and Sergio. And then I think we'll keep the final question for Mark Peterson. What are some lessons learned in terms of the interaction with the FDA on the use of QSP modeling to inform the dosing regimen exploration for the label?
Thank you, CJ. So I'll have to speculate here a bit because I didn't work for MPS, but I did have the pleasure of speaking with FDA representatives Nitin about this. I would imagine what was learned is there's an opportunity in these situations to use QSP models to flesh out what you've done in clinical study with respect to the dose response. And so hindsight being 20/20, I imagine MPS probably could have, if they were aware of the bone and calcium model, done that work a priori to either inform their trial designs or to use it to supplement what they had going into the filing. From the FDA's perspective, I think their view is that there are definitely places to use these models in the way that I've just spoken about, and probably others. We could probably all envision a number, but in the interest of time, I'll just say that it's probably the case that they won't usurp the traditional approach where you've got a fairly robust dossier. And I think we should see these as complementary tools, both as we've seen here today as hypothesis-generating tools, as well as bolstering our understanding, perhaps moving labels from mechanism not known towards something that's viable based on the totality of our understanding of physiology. And that would be nice to see in the future.
Okay, I agree. Great. Well, thank you very much. I'd like to thank all of our panelists today, and I will now hand it over to Alexis from the IQ Secretariat to close the session.
Thank you very much, CJ. So I first want to thank all of our panelists and presenters for a wonderful presentation today. And I want to thank everyone who took an hour out of their Friday to attend this webinar. If you have any more questions following the webinar, please send them to Maha Marshall or to Alexis Myers at the IQ Secretariat. That is majarshall@dbr.com. And we will ensure that those questions are answered. So with that, I want to thank everyone once again and wish you all a wonderful afternoon.